Gordon is a physicist by training. He's an assistant professor of biology and a member of the physics graduate program at Emory University. And his lab uses theoretical and data-driven approaches to study repertoires of animal behaviors. So the primary focus is on the temporal ordering of behaviors, their control, and the evolution of an organism's movement. Gordon, take it away.
Great, thanks, Anne, and thanks everybody, Anne and Pietro and all the other organizers for putting this thing together. It looks like it's going to be fun. So I'm going to talk about a very small subset of the overall stuff that people here are probably interested in. And as Anne mentioned, I sort of have a physics training masquerading as a biologist at the moment. And so really what I'm going to talk a lot about here is focus more on this question about representation, this question about how do we go from raw data of animals doing what they're doing, and how do we go and try and generate a representation of behavior to answer specific sets of scientific questions. And so I'm going to be a little bit more precise about that in a moment, but let's just get in. So what we're interested in my group, so we have a theory and computational group, and so we're interested in this broad question about how do we put numbers on these quantitative and these really complicated types of activity that you're saying all these animals behave. And how do we actually try and make sense of these in a quantitative sense that allows us to make predictions about their underlying neurobiology or genetics and evolution and all sorts of interesting things like that. And one of the things that I definitely don't need to mention to this particular crowd, including the stuff that Ian just mentioned, is we've made a lot of really great progress over the last few years of using various deep learning based techniques to go and track either maintaining identities for large numbers of animals or tracking specific postures. But the really sort of the question that me and my group have been really interested in is not so much sort of the instantaneous tracking or positioning of the animal, but more sort of along the lines of what Ian was talking about, is what are these kind of internal states? You sort of, how does an animal make decisions? What are sort of the subtle underlying things which bias how an animal decides to go about the various things throughout its day? So on one level, there's something like circadian rhythms, which over the standpoint of 24 hours, you can see different biases in terms of relative amount of activity or hunger or social interactions in parenting or aging. And we study various aspects of all of these things in the group. And one of the things sort of we like to think about is how do these various internal states like circadian rhythm or hunger interact with each other? Say, for example, like if you're laying in bed at night and you're hungry, do you decide to get up and eat, right? This is sort of, you have this war between these internal states, they're interacting, they're playing against each other. But these aren't things we can directly measure, right? We can measure an animal's ability to eat or desire to eat, what it's willing to do to overcome particular obstacles. We can't measure this internal state of hunger directly for the behavior. So it becomes an inference problem. And the question is, how do we infer these longer timescale structures, and how do we make predictions then about how to go from behavior down into these lower sort of more mechanistic levels? And so that's what I'm going to talk a bit about today. And so the picture I have in my head, and don't take this too literally, but this is sort of the picture that I sort of think about a lot of the time when trying to understand these long time scale states, is imagine kind of we have an animal sort of wandering through this kind of dynamic warping landscape where each one of these sort of valleys here might be a stereotype behavior, like grooming my head or something. And there's some amount of variability within that. And then there's some noise. And so you're sort of hopping between these various types of things that the animal can do. Now, but what can happen with time is that the landscape might warp or move. And so that might change the variability in a behavior, that might change how you're going to move between behaviors. And so then understanding this feedback, which is usually due to changes in internal or sensory state, we might get the sort of feedback. And so the question then is, how can we kind of understand or try and approximate what these functions might be? And how can we then use that to make an inference about what some of the interesting internal states of the animal's behavior are? We're not going to get there today, but this is kind of, I want to show this as kind of the underlying picture of what our big goal is and what we're trying to do across a variety of different projects in the group. And what we're going to focus on particularly today is we're going to start from this type of representation. How do you represent sort of the static picture of the behavior that animal does? And then how can we then sort of warp that to see what might be happening over longer time scales? And one of the things that sort of is striking if you look to see what's happened throughout most of kind of the more modern behavioral literature is when people model kind of sequences and structures of behavior, how an animal goes from one thing to the next thing, to the next thing, to the next thing that it does, there tends to be this very Markovian picture. So it means that essentially what an animal does is only dependent on essentially the last thing it was doing. Maybe it can be a little bit fancier by adding in some autoregression on some shorter timescale, but really what this effectively means is you only have about 1 second of memory-ish in your system when you're describing it. And we all know from sort of our own intrinsic sort of note of how we behave and watching our dogs or other animals behave that there's really are much longer timescales that exist in this. And so these are a bunch of really cool papers that that I think do a lot of interesting things, but really what we're trying to do is move beyond. How do we actually predict these behaviors, moving from this kind of scale of individual actions up to longer sequences? And where there's something interesting is, of course, the ethologists have been thinking about this for quite some time, for folks like Tinbergen and Dawkins and Heiligenberg, all really sort of have had in different sorts of ways thinking about how do you build structures out of more simple patterns. And really, so half jokingly, a lot of what my group does is take a lot of the ideas from classical ethology and tries to put more sophisticated math and data analysis techniques on top of them to try and sort of make sense of these, a lot of the brilliant ideas that these folks have. So let's sort of get in and sort of ask this question about, all right, so what do we need to build this representation? Really what I'm going to claim is you need 3 things. We need a means of representing short timescale behavior that permits moving between timescales. So that means that this sort of Markovian picture doesn't sort of work super well, because we'd like to have a way of describing the actions that animals are performing in a way such that it's not necessarily dependent on our definition of that behavior was not sort of defined on this 1 second timescale precisely. Then on top of that, what we need to do is we need to now have a way of going from that short timescale structure of these stereotype behaviors, and then actually have these coarse and fine graining procedures to see sort of how we're going to move between the timescale of hundreds of milliseconds to seconds to minutes to hours to a lifetime. And then 3, what we need to do is sort of, and really this is where most of our group is spending a lot of time now, is developing theoretical infrastructure to create predictive models that link behavior to all the stuff underlying it. And by predictive, I mean the future of that same animal, and other experiments where we do different types of perturbations to the animal or other modalities. So for measuring behavior, you can make predictions on, say, underlying neural activity or on how sort of behavior might have been evolving. And so let's start with 1. So 1, this is stuff that we've been doing for a while, but really what I'm going to claim is a good way of doing this is looking at stereotype behaviors. So what do I mean by stereotype behaviors? Again, a physicist training. And so imagine this is the space of all possible postures an animal can make. It could be, say, like the Euler angles of my arm here. And there could be some simple movements. There could be some periodic movements. This could be some completely crazy things, or even some nutty stuff that fill up the entire space. But if we watch an animal behave, what we typically see is something that looks more like this, where we see the same behaviors performed often and repeatably. And we're going to call these our stereotype behaviors. And so then what we're going to do is watch to see how the animal moves between all of these sets of stereotype behaviors and this kind of meta dynamics, and hopefully that this can lead us into some sort of insight. So we have our way of identifying this. This is rather old at this point, so I don't want to go too much into the detail. I'll point out that there's code available for these things, both in MATLAB and Python. But the key factor here is that what we're really doing is we're taking time series of what the animals are behaving. So say, for example, those tracked postures that I showed you a few slides back, and then we're looking at using wavelet spectra structure to try and isolate sort of in a multi-scale way what parts of the animal what points in time are the animals moving similar parts of their body at similar speeds. And then there's stuff we add on top to try and embed it into this 2-dimensional space so that we can isolate those points in time together. So doing this for the case of Drosophila melanogaster, so these are a bunch of flies that we just put in and filmed in a relatively featureless environment. Here's the type of what we call our behavioral space that we create. So for example, we get a bunch of points in time that map to this peak here. and take random selections of what the fly is doing, they're all running. Or up here, the flies are all grooming their head with a slight left asymmetry if you look carefully. Or down here, the flies are all grooming their left wing. So we call this our behavioral map. There's a structure to this where, again, similar point, in this map, similar points in time are kind of put together if the fly is moving similar parts of its body at similar speeds. So here's idle, here's like proboscis extension, various types of running from fast to slow, anterior grooming, posterior grooming. So we can build this type of representation, and then each of these peaks kind of can be our representation at the shorter time scale. And not to belabor it too much, we've done a bunch of other more recent papers where we're talking about stuff in other animals. So whether it's in rats, in this recent paper, or in humans, stroke rehabilitation, or looking at stuff across multiple time scales. And I'm happy to chat about this more directly later. But okay, so we have this representation at the shorter time scale. So then how do we move from these shorter time scales out to longer time scales? So how do we define a coarse graining procedure? So if we take this map, again, these are flies. And what we can do is say, given that I start at one of these peaks, where's the next peak I go to? And we can create a transition probability structure throughout this map. So each of these lines correspond to the Markov transition probability of going from, I start at this state, what's the probability that the next state I go to is that one. And the handedness is the direction of transmission. And what you can see from this is it's not a hairball, right? Most of these transitions are kind of nearby in the space. So this is kind of nice. So what that means is the space really, so the fly tends to move to similar types of behaviors. And so that allows us to then explore a little bit more. And we can ask the question, well, I said before that maybe this Markovian picture might not be so good. Using this framework, we can actually test that. And sure enough, we can look at the transition matrices, say between what's the next state I do, or what's the 100th state in the future I might do, or what's the 1000th state in the future I might do. And what you can see, we can quantify this based off of the eigenvectors, the eigenvalues of this transition matrix. And here, this dashed lines are what we would get for our Markov model. And these lines are what we get for our actual model. And we can see we get the actual data shows much, much longer timescales for a bunch of these eigenvalues. And so this, as it turns out, if you actually try and write down a model that does this is quite hard. However, but this is telling us there really are these long timescale structures. And so what we can do is say, okay, how would we want to describe the space? How would we coarsen the space out in a way that would optimally preserve this long timescale structure? And it turns out what we can do this through by minimizing this functional where we say, let's preserve the information about the future. How well can I predict, while at the same time minimizing sort of my representation or making it as simple as possible. And so we can move along sort of one of these curves where down here is a simple model and out here is a complex model. And so this model doesn't predict very well, but this model predicts well, but it's more complicated. And as we move from simple to more complicated, what we see is the space breaks down hierarchically, just like sort of the classical ethology models of Tinberg and others. So we have this notion of hierarchical structure and behavior, and that we can actually predict what are the optimal coarse grainings of the behavior, describe behavior at longer and longer timescales. And it turns out this actually allows us to write down some nice models and that I won't have time to talk about today. And so lastly, and I'm just going to talk about this in the last couple of minutes that I have, is talk about sort of the theoretical infrastructure. So one of the things that we've been doing that I won't have time to talk about is really how do we then use kind of modern machine learning methods to create sort of these simpler sets of models that can reproduce the types of patterns and sequences over long time scales that we see. So I'm happy to chat about that either in the questions afterwards or later. But what I want to talk about in the remaining time is a surprising sort of correspondence that we came up with when we started to look at the evolution of flies. So we have Jessica Candy, who is a former postdoc with David Stern at Genelia, took data from about 600 flies from 6 different species of in the Drosophila melanogaster species subgroup, and that we were able to make different kind of behavioral maps for each of the individuals within the species and for each of the separate species. And if we then create a map sort of where we're putting similar types of species next to each other, we can see that there's each of these different colors here is a different species of fly. And we can see that in this kind of abstracted space that it's not completely random, that there seems to be some structure, that the behavior actually is a trait that we can try and map. And we can actually show this by then just doing a very simple logistic regression that we can get above around overall 90% accuracy with trying to predict which species it was if you give me the behavior. And so what we did, and really this is the work of Damian Hernandez and Catalina Rivera, two very talented postdoc and grad students who were working with me on this. We were able to basically map how behavior was changing across this phylogeny by essentially fitting two covariance matrices. One, a covariance matrix, which is how things are moving together, and another one is how we would predict behavior to stay the same across within a species. And what's really interesting, what I'm going to focus on here is the second part. And it turns out if we measure the individual covariance matrix, so these are the probability of performing certain behaviors more or less within the same species, we're essentially get identical clustering or coarse graining of the space that we would have gotten from this type of coarse graining procedure that I talked about previously on just the Drosophila melanogaster. And so we get this type of correspondence. Really what that's telling us is that there's a weird sort of ergodicity that we're measuring, and that essentially a lot of the variability that we're measuring across behavior, what looks like variability that we're measuring when we measure multiple animals, really might just be the variability of these internal states. And so by basically averaging across individuals within a species winds up being similar to averaging over time, which is sort of at its core kind of this definition of ergodicity. And so this has sort of led us to sort of a bunch of interesting things of how we could then go back and infer these internal states and how we could then maybe correct for some of the sort of additional variants that we're seeing in the data when we're trying to map genomes across the different species. And with that, I just want to end sort of with this picture and thinking about kind of how we can then go and see how we can predict how this, what are these internal states, how this sort of map is warping with time, how animals are moving between things and things, and how we're sort of building across in a variety of systems in the group from flies and rodents and all the way up to humans, trying to think about what these structures happen. And I will just thank the great people that I work with, us all hanging together in pre-COVID times. And I'll also note, and all our great collaborators, the folks that give us money, and I'll also put in a shameless plug that we're currently recruiting both postdocs and grad students in the group. So if you're anybody curious, feel free to chat with me, and thanks.
Okay, thank you so much, Gordon. We have some time for questions.
Yeah, I have a question. I'll ask it because I'm about to step out of the train and I want it to be on record. So, Gordon, how do you, maybe you said it, but I didn't get it. When you cluster different actions, there is the usual problem that some actions are very brief and iconic, like wing extension. It takes roughly always the same amount of time. And some actions like chasing or walking or grooming can take highly variable amount of time. So you have you have a long tail distribution of durations. How do you deal with that?
So first of all, that's a great question. So I would say there's two separate timescales of interest here. One is the timescale of the behavior that's actually being performed. So there's the timescale, say, of locomotion, of the timescale of the limbs moving. And then the second timescale, as you mentioned, is the timescale of how long do you stay running. And so what this approach does is it decouples those two a little bit better. So what you have is the timescale of sort of what defines the running behavior is the timescale of the action the animal is taking. So the timescale that it's moving its legs. But the time scale of locomotion is then set by basically how long do you sort of stick in one spot in that space. And so that somewhat decoupled those two. Now, you mentioned other behaviors such as chasing. So chasing is actually a combination of a bunch of different stereotype motifs. And then how do you act? And so this to me gets more to this kind of coarse graining type procedure and something that we've been actively thinking a lot about. And so I'm at a workshop right now, and so Greg Stevens, who's another behavior researcher, and I have been thinking a lot about kind of how do you take these kind of detailed time series of animals interacting and sort of isolate these epochs in time, which might look like which are multiple sort of behaviors stitched together, but really they're a combination of different things. So that's really sort of how we think about it. But of course, there's always a little bit fuzzy because behavior is so multi-time scale.
I have a question about the end of your talk. You showed the phylogeny of Drosophila species, and
a very, very narrow one, like only 6 species.
Yeah. But you got this divergence in behavior that you were saying was kind of a consequence of just, like, I guess, the behavioral drift over time. Is that the kind of argument you were making, that, like, if you were to separate into, pan-mictic populations for, X amount of time, then you'd just end up with this same kind of pattern of behavioral divergence?
So, I don't want to make too much of this. Again, this was more like what I was showing there is kind of a result of, this is a way of analyzing repertoires of behaviors across multiple species. I don't want to necessarily make the direct claim about whether these were evolution, whether this was neutral, or whether there was any selection involved or anything like that. All I'm saying, we made a neutral model where we have kind of variability within a species, and then we have a random walk effectively on the phylogenetic covariance. And then we do a Monte Carlo simulation, or many, many Monte Carlo simulations, to then try and match those endpoint distributions as best as possible, starting from these various things. So that's really all we're doing. And then we're basically, the key thing is, okay, so now what do those covariance matrices look like, and how can we learn anything from the structure?
But you could potentially shift your analysis to behaviors that are, thought to be adaptive, and also behaviors which are convergent, to then see if they break the pattern, to see if there is this kind of reticulation of the behavioral tree in species which are phylogenetically independent but have similar behaviors, at least, a priori, you think that they're similar.
Yeah, no, and certainly in things like courtship, we have every reason to believe that there's a lot of adaptive behaviors and other sorts of interesting things. This was kind of just our first stab at trying to make some sense out of all of this. But yeah, certainly those things could be they're all testable hypotheses with this type of a framework.
Thanks.
We have a good question from YouTube. This is Ralph Peterson, who says, thanks for a great talk, and wants to know what we can expect to learn if we increase the resolution of posture being fed into these models. So if we add more key points or higher frame rate or 3D pose estimation, are things going to change?
Oh, well, that always depends, right? So, for example, when I was showing you a bunch of stuff in flies, and we're filming at 100 hertz, it's great for watching flies walk around in a dish. When I was a grad student, I was doing insect flight, in which we were filming with a phantom camera at 8,000 frames a second, and the most important thing that happened over about 4 to 5 frames each wing cycle for that. So to understand the high-speed aerodynamics, we needed 8,000 frames a second, and so that, to a certain extent, yes, you are going to discover different things at different frame rates. And so that's always sort of goes into these choice of timescales that one always needs to make. I think we shouldn't hide from the fact we're making a choice of timescales always. When we're making these analyses, we need to think very hard about how to do it, and then we just need to say what we did, and then people can argue with us. But you're always making a choice. You can hide it if you want, but you're always making a choice, whether you're hiding it down into some algorithm or not. So that's one thing. The other aspect is about sort of the deep learning based tracking and other sorts of things. So I will say for the flies, actually, we've done this on Leap/Deep Lab, got tracked wing angles and everything else on the fly. You get essentially an identical map out. So for the flies, it winds up being not a huge deal, partially because they're both literally and metaphorically very rigid, and that there is just a lot of structure in their movements, and they tend to be very kind of almost robotic and repeating stuff over and over and over again. When we do stuff with rodents, the representation and how you do the tracking winds up mattering a lot more, and we've had to put a lot of thought into that. And again, I'm more than happy to chat at various points about that, but that's probably a much longer conversation. But it can matter. The long and short of it is it can matter, but often it doesn't.